I'm the marketing manager here. So I've been working in biopharma industry for more than 10 years. So today, I would like to take this opportunity to share with you about how Gyrolab platform used in ADC analysis. So let me share my screen. Okay. So the topics I'm going to cover today would be like those ones. Um, so the first one, I would like to talk about the challenges in ADC analysis. And then after that, I'm going to introduce something about Gyrolab briefly. And then we will focus on the application studies that we have been worked with our customers for ADC PK analysis. Okay. So I think for antibody drug conjugate, everyone is quite familiar because this is a really booming topic among in the biopharma industry. Because this drug, it's not really, it's more complex, like more than like monoclonal antibodies. It's more, it has three parts: antibody, linker, and payload. And since this molecule is more complex, and also its mechanism action is also more complex because it will target the tumor cell by the antibody part. And then when getting into the cell, the small molecule will be released and then kill the cell. But the small molecule will also infect the environment of the cell and also get into other cells. So this is a quite complex process than other molecules. And also because of these molecules, they have different DARs, meaning they have different drug antibody ratio. So the small molecule, how many small molecules will be tagged into linked to one antibody, that will also make it a little bit different on the analysis part. So because of its complex structure, and the analysis part will also, like PK, PD, and ADA, will also a little bit different than other molecules. So first thing, it need to analyze the total antibody, meaning it's not only the antibody that has linked to the small molecule, but also those naked antibody that has no small molecule or payload on the antibody itself. So the total antibody will be tracking and monitored, and after dosing to an animal or to a patient. And another analysis is focused on the total ADC, which is intact ADC concentration, meaning only those antibody have a payload will be detected. And then also about the small molecules part, like the payload part, will also need to be analyzed. But for this part, it's usually using the mass spectrometry analysis platform. And then lastly, the ADA, as other biomolecules similar as others, it also needs to be tracked. So that means in ADC, we have more assays or methods need to be developed. And thus, there will be more reagent needed or relevant to this method. So the first challenge is that there will be multi-reagent required to set up a good assay. And also for this reagent, it may have the anti-payload antibody, or it is the anti-specific antibody, because the anti-payload antibody will be used for the ADC or intact ADC analysis. And the anti-specific antibody will be more important for the total antibody analysis. And for these of reagents, sometimes it is quite specific reagent, so may need more longer time to produce. Maybe sometimes it will take around 3 to 6 months to produce and validate. And also that will be leading to the timeline constraints because sometimes we need to meet with our R&D timeline and to speed up the assay development. But these could be challenges to make it more difficult to meet that timeline. And additionally, we also need to prepare more samples and internal standards and calibrations and positive controls to set up the assay. So how to solve these challenges using Gyrolab. Here, just to briefly show the benefits of Gyrolab in ADC analysis, but I think it would be better that I can explain in the later technology introduction and also the application study. But to briefly talk about that, Gyrolab is quite a matrix tolerance, so we can solve a lot of matrix effect issue. So that will benefit for those complex samples like animal tissues or homogeneity, or it also benefits the low affinity molecules, or even you use the low affinity reagent. This will help you solve the problem. And then because this is a automated platform, so we could support very very fast reagent screening, and then we could also help you to resolve if there's limited reagent, especially for those specific antibody when there are limited amount, or you want to reduce the cost of the reagent that using for the bioanalysis. Then this platform is quite useful because we don't need to really consume a lot of reagent and samples. And lastly, is the sample volume is quite low. I'm going to share with you more details about this technology before we really get into some case study and data sharing. To give you an idea of how this platform works and how it can solve the problems we just talked before. So this technology, I'm not sure if you have heard it before, but it was set up in around 2000 in Sweden. So it's a Sweden company. Everything like instruments and also our consumables, we produce and research them in Sweden. And this technology actually comes from Amashare, which is previously part of GE Healthcare. So then it was separated and set up the new company, which is called Gyrolab은. And we have our first instrument, which is this one. It's called XP, which is a 5 CD workstation. It has very high throughput, quite quick. And because we also want to support some customers which they may not have too many samples to analysis, but still want the speed and automated platform. So this one is a smaller one, which has only 1 CD inside. And after that, this company also acquire another company, which is called Protein Technology. And this one is focused on peptide synthesizer. And now we are part of MasterLab because MasterLab acquired Gyrolab은 in 2019. So now we're part of it. Okay, to talking about the basics of this technology. So you can think it more simply like it's the basic methodology is based on LBA. But the work, let's see, mechanism is not quite same. So we know, I guess you all are very familiar with the ELISA platform. So it's a plate-based platform. and you also need to pipetting manually. And but still, there are also commercial kits for this platform. Gyros actually, the basic is still also based on LBA, but we are based on we will work on this CD, if you can see it. And this CD is a microfluidic technology. So meaning that everything on this CD, no matter the samples or the reagent you put in, are quite limited number. So it's nanoliter volume. And it's quite automated platform. So what you need to do is just place the samples in this 96 plate. Similar as what you do before, and also place the reagent into another plate. And after that, everything will be done automatically. So the samples and the reagents will be added into this plate automatically by needle. And the speed is quite fast. So for ELISA, it may take around 3 to 4 hours to do one plate analysis. But on Gyrolab, this one CD, we just need take 1 hour to get all the results. As you see on the last page, the CD is quite important. So this is our high core technology. If we maximize some part of it, we can see there are some microfluidic channels. So the reagent, like the antibody, will be added into these common channels. And also the sample will be added into this part, which is a volume definition chamber. And the core part of the CD is this affinity column, which we can see it like this one. So there are a lot of beads, resins inside this column for this affinity column. And it has 15 nanoliter. On this beads, we have the biotin labeled antibody, and then you can add the analyte, and then the secondary or the detection reagent will be labeled with Alex Fluor 647 fluorescence. So after each of these analysis, so this is one data point and one data point. After each of these analysis, the data point will generate like this column profile. It is fluorescence signals. Using this profile, it is very easily to justify whether our reagent, the detection and capture reagent, has a good affinity with the analyte. So if we can see here, this is a very good profile. It has very sharp peak. If in this way, meaning that we have very good affinity, but if it is like a tagged, long-tailed profile, then that means the affinity is not so good. So this profile is quite easy for us to justify the affinity, but also we will usually use it for like troubleshooting because if you see different shapes of this profile, then you can justify where the problem is and then adjust your method or your assay or your reagent. And the dynamic range is also quite good. So we usually have 3 to 4 logs, and sometimes we can achieve 5 logs, which is much wider than ELISA platform. Now, I'm going to show you a video that maybe better explain how this process on Gyrolab.
Let's take a close-up view of the CD. The biotinolated capture reagent is added to the common channel and drawn into the microstructures by capillary action. Hydrophobic barriers stop the flow
This is
Then,
a
with a higher
hydrophobic
speed of the
barrier, so
CD,
it will not go
centrifugal
through,
force
but
drives
when
the bioaccumulated
the
capture
CD
reagent over
spins,
the streptavidin coated bead column, and the capture reagent binds to the beads. The needle overflows the samples, which are drawn into the volume definition chamber, and a low-speed spin results in any excess volume
this is
being
the
eliminated
chamber for
at
sample.
the overflow channel. Volume definition drives our excellent reproducibility, robustness, and ability to rapidly cross-transfer assays. With a higher spin of the CD, the sample enters the affinity capture column. The fluorescent labeled detection reagent is added through the common channel, and the centrifugal force drives the detection reagent over the columns. At this point, the sandwich immunoassay is completed. The internal laser scans the 112 columns in 90 seconds. One CD delivers 112 data points in about an hour.
So that's the whole process of how it analyzes the sample. So all the things you see is automatically handling by a needle and an arm chamber. So all these channels, the only thing that didn't explain here is that once every sample was added, there will be procedure that to wash the needle to reduce or to eliminate the contamination risk. And the platform is quite open. So we have some ready-to-use kit, but also, for example, like if you want to develop your own method, it is quite open. So we have no limitation on the reagent. Theoretically, what you have done on ELISA platform, that could be done on this platform. So the reagent and the assay could be transferred here. And we can support different method formats like sandwich assay, PK, bridging, and indirect assays. So there's no limitation to develop on this platform for assay. And here's just a brief about the product itself. We have two types of the instrument. So within this instrument, you can place 5 CDs, and meaning more high throughput feature. And within this one, there's only 1 CD. Usually for CRO labs, they will have this one because they have lots of samples to analysis. Then 5 CD is quite fast. Every day they could maybe do more than 10 CDs per day. That means because 1 CD we have 96 data points or 112 data points. It depends on which CD you choose. We have different CDs to support with different sensitivity or dynamic range requirement. And with this one, it's more commonly used among our pharma customers because for some pharmaceutical companies, if they just focus on assay development and then transfer the assay to CRO for sample analysis, then they may not deal with a lot of samples. Then this 1 CD system is quite efficient for assay development. But I know that also some pharma companies, they have their own group for sample analysis. So if in that way, then this 5 CD is more applicable. And we also have a lot of consumables, software to support you with different functions. And our service team is also, we have 7 service engineer based in APAC region, so that can support you better. Also our partner, Coma, they will also, they are located in Korea, so they can support you very quickly. A very quick look at the inside of the CD. So it's of the platform. So here we place the CD, and here the two plates, one for the sample and the other one for the reagent. And one thing to mention here is that for the reagent, you don't need to place your reagent, distribute your reagent into each of the well. You just place one the capture reagent in one well and the detection reagent in another well. That's fine. But the amount should be applied for the full CD. So other things, the needle will take care of the reagent distribution on the CD platform. Okay, so we actually cover lots of applications here, but today maybe we just focus on the PK part. If you are interested in other applications, we also have other case studies and publications. So if you are interested in any of them, let me know. I can share with you. So I'm going to share with you some of the advantages. This is just a very brief introduction. From time consuming point of view, we are quite efficient. So one CD, 1 hour. We don't need to invest a lot of time on the platform operation. So comparing with ELISA platform, we are quite efficient. And also, from sample and reagent consumption point of view, each of our CD, or let's see, you can regard it like a plate. The CD has 96 or 112 per CD. But the sample volume we needed will be around 4 microliter to 8 microliter for each of the well. So quite less than ELISA, because ELISA maybe sometimes they need around 100 microliter. And for the capture reagent, for the full CD, I mean the full plate, we just need 4.6 microgram. And then for detection rate, it's much less. It's only 0.2 microgram. So that will be only 1/10 of other platforms. In this way, the most applications benefits from this point are those ones using like transgene mouse, which means the mouse is quite high cost. Then in this way, we could really reduce amount of mouse that we use. Why is this? It's because we could support microsampling. So for each of the data points that we collect from mouse, usually like ELISA or other platforms, they need at least 200 microliter from each of the mouse. But if using Gyrolab, we just need only 10 microliter per data point. That's already enough. And this is from a scientific or cost or ethical point of view, it is quite beneficial. Just an example to save mouse here. So if we can support microsampling, then for one mouse, we can take 10 data points per mouse. So meaning that with one mouse, we could create a full or a complete PK profile. But if using other platforms that need to consume more samples, then you can't use only one mouse for all the data points, because then the mouse can not support so much sampling. Then you need to use a composite sampling way, meaning that you need to take data points from different mouse to create one PK profile. And that will generate more needs on the mouse, on the number of the mouse. That will be 6 times more than what we can use on Gyros. So for example, this is 3 mouse, and this is almost 18 mouse that you need to use, 15 mouse. But also this will also benefit for those drugs that are quite costly. like gene therapy or cell therapy, which the manufacturing cost is quite high, then we could also reduce the sample needed. As well as those reagents that are quite costly, then you could also benefit from this. And then this is a very special benefits that we have comparing with other platforms, which is reducing the matrix effect. Why it is? Because if you see from the video, all the flow is coming through the column, I mean the affinity column. It has very the contact time is very limited, so less than 10 seconds. That means in this short time, we can ensure all the things that we want to capture could be captured very quickly. But for the other like interference matrix, then we let it go through quickly. We don't need to incubate at this time because the column is quite the size of the column is quite small, so it's only 15 nanoliter, and the sample and the reagent is also very volume is quite low. So we just use very limited time to capture what we want to capture, and then let those interference things just go through and do not interact with our samples, with our analyte or reagents for a longer time. Like in plate-based platform, we need to incubate a longer time. So that will also increase the risk that your matrix will interact with your analyte or with your reagent, meaning the signal you get is not only your analyte, maybe it's from the matrix itself. This I'm going to share with you the case study later on that really benefit from this point. As a must in automatic platform, so our data precision and reproducibilities are quite good. Usually we will less than 8. The CV will less than 8. And sometimes if the assay is quite robust, we can also achieve less than 5 CV, no matter it's inter CD or in between the CD. Here, I would like to share with you the first case study, which is a biospecific ADC. So here is the background of this project. And it has now it's a biospecific CD with DAR4. And it has 2 targets. So one target is PDL1, which is only 19.3 nanomole. But the other one we just haven't disclosed it here. But the affinity is not so bad. It's at a good range. But the PDL1 part it has very low affinity here. At first, this PK analysis was done on ELISA platform, but according to their regulation feedback, they need to capture both the 2 targets to do the PK analysis. But on ELISA, they can only capture one from NTFC from this part, and this the other one capture this part. And this method was rejected because they cannot accept this because they also get previous challenge from the regulatory. So that's why they tell that they need to capture the 2 parts, the 2 targets. But after that, ELISA cannot do that because they cannot set out the standard curve. So that fails. And then they transfer this method, try this method on MSD. But it was faced with a lot of random CV issues to capture the both target. And concern from the customer side is that maybe it's because the PD-L1 has quite low affinity, and that will be easily to be influenced by the matrix effect. So because they also have Gyrolab in their lab, and they know that Gyrolab could solve a lot of matrix effect issue, then they transfer this method to Gyrolab to have a try. Yeah, we have got the full data, but according to our customer, they don't want to really show all of them. So I just selected some representative data here to let you understand how the platform performs. And the standard cover are quite good to target two targets, one PD-L1 and the other target. So we use the same capture and detection reagent as with MSD platform. And we don't see much CV issue here. And the customer, actually, their needs is that they could accept a higher level of sensitivity, meaning they don't need to really like nanogram sensitivity. So 1 microgram sensitivity, they are satisfied. But the only needs they tell us is that they need to keep the CV really, really low or very stable. They don't want to see the random CV issue again. It's better that we could achieve less than 5 CV, 5% CV. And we give a try and use the same assay format, and the data are quite good. So with the 1 microgram milliliter sensitivity, actually we could do that lower because the signal to noise is still more than 10. So actually we still can reduce it or lower it, but it's already meets the customer's needs. So we just keep it here. But from the CV and bias point of view, it's quite good and quite stable. And they also do the similar QC samples with DAR2 because if you see this molecule, the DAR is 4, the DAR is 4, and they also test the DAR2 samples as QC, and the data are also quite good. So there's no DAR sensitivity issue, and all the CV are less than 5, and that meet with their needs. So finally, they choose Gyrolab for their clinical phase 1 study because if they see random issue on other platforms, it's quite risky to move forward with their clinical study sample analysis. And that's why they want to select a more robust platform and also robust method on Gyrolab. And usually, recently, we also see some other customers facing similar issue. Sometimes it's because of the low affinity of the molecule itself, or sometimes they don't have very good reagent. For example, they only have the commercial available antibody to support the assay, but this always sometimes happen in the preclinical study. And the commercial available reagent is, although it's easy to get, but sometimes it doesn't have a very high affinity with your samples. So that means sometimes the assay is not quite robust and data are not quite good. In this kind of cases, Gyrolab, we could support it well, better than other platforms because sometimes the low affinity issue problem will be let's see, maximized by matrix effect. So if you solve the matrix effect issue, then the data could be better. Okay, then the second case study I would like to share with you is the PK assay development on it's not a bispecific and ADC, it's just a simple ADC. But this company, they have a lot of ADC pipelines. So meaning they need to do a lot of assay development and also need to do a lot of reagent screening things because they need to find the right format and the right candidate for their robust assay. So usually the ADC PK assay need to screen a lot of reagent, especially when you have a lot of pipelines. And the challenge is that they need use a lot of time to screen maybe hundreds or thousands of antibodies to choose the right one to develop your assay. Sometimes customer will use ELISA to screen, and but Gyrolab here could also helps a lot and quite faster than ELISA. Here we just listed out the clone selection process. Usually we will start with the supernatant screening and then identify the best candidates and then confirm the specificity because we don't want to have the cross reactivity in different animals or in between the patients. And then we need to optimize our capture and detection combinations or format for the best performance. In traditional method, usually it takes around months, maybe 3 to 6 months to screen all these reagents. But on Gyros, actually the screening is quite fast. Why it is quite fast? We can see from this case. On one CD, you can, if you maximize it, there will be some different segments here. In each of the segment, there are 8 data points. And for these 8 data points, you can use one pair of antibody, meaning one capture and one detection reagent. And in the other segment, you could use a different pair of reagent. So meaning on this CD, for example, this one, this is a 200 CD, so it has 112 data points. And then it has 14 segments on that. And for these 14 segments, we could use 14 antibody pairs. Meaning, and so if you use this one CD to screen, then you can have 14 antibody pairs screened within 1 hour. And here, this case is to screen 20 antibody pairs. Then they use only 2 CD and within 2 hours to finish all this screening. And although the time is quite fast, another good point is that if we use ELISA to screen different reagent pairs or antibody pairs, you need to pipetting different pairs of reagent manually. So meaning you need to combine do the different combinations by yourself. But if using Gyrolab, actually you just need to place your maybe 5 or 6 different antibodies into different wells of the 96 plate. And then the platform or the needle will help you to combine different antibodies together into these different segments. So you don't need to manually to combine them. And it's the platform itself, the instrument, will combine, do different combinations of the antibody pairs. So that will save a lot of manual operation, but also reduce any, let's see, operation errors, because you can just modify which 2 pairs of the antibody you want to get together in the software directly. And also the good thing to get your results, one thing we could have the standard curves like this to justify whether this is a good or not good antibody pair. But also you can use these fluorescence profiles. Just like I mentioned before, you can just quickly define how your antibody pair with your analyte the affinity is good or not, justify from these profiles. So this one very good affinity, and this one is a little bad or low affinity binding with your analyte. But one thing here, this is not a it's not a quantitative analysis. If you want to really compare that with like SPR technology, it's not the same. So we are not really analyzing like dynamic affinity analysis, or it's just like a final data of affinity. Okay. Yeah. Okay, so except for that, so this is from Daiichi data that they have developed different assays for their ADC platforms. So like for this one, using ELISA, they will do, for each of the assay, it takes around 6 months from reagent screening to assay setup. And then for each of the assay, they need to do on because they need for preclinical study, they need to do on 2 animals, and for ADC, they need to have 2 assays, one for total antibody, and then the other one for total ADC. So it will takes around 1 year to finish this project only. But on Gyros, it just only take around 1.5 months for each of the assay. Usually the assay development is quite fast. It's only 1 to 2 weeks. And then combining with assay transfer and assay validation. So it's quite fast. And then 2 animals, 2 assays that will consume only 2 months for each of the project. So that's quite a fast time saving. And not only the time, but also the consumption of the samples and the capture and the detection reagent. So we are using quite less than ELISA platform. To just quickly summarize, the time reduction is quite fast, more than 90% reduced. And then sample volume, we just consume less than 5 microliter for each of the well, and the reagent saving is quite optimal. Okay, to summarize where we can do better in ADC analysis, the one best or popular reason that's why customer would like to use Gyrolab for ADCs because the low matrix effect, or it's quite good matrix tolerance. So in that way, we could support different samples, complex samples, as well as some of the low affinity molecules that may have been easily induced by matrix effect, influenced by matrix effect. Then thus we could get more reliable data performance. And another thing is that because of its low matrix effect, we could also support on the commercial available reagent, which also have a little bit lower affinity than the specific ones. So in that way, if you didn't have a specific reagent right now, you want to use a commercial available reagent, that's fine. We could also deal with this. And with this good benefit, sometimes you can save your timeline because commercial available reagent is more easier to get. And also because it's quite fast data read and also automation platform, we could accelerate the assay development timelines from comparing with ELISA, we have better, almost 90% saving on your time. And reduce the sample and reagent consumption. That will be more beneficial for those transgene mouse PK or some gene therapy, and as well as some eye disease drugs that use for eyes, because the sample is also quite limited in that way. And so in short, so Gyrolab delivers really high efficiency, but also reliable data for ADC analysis. So we also have different offices worldwide, and our R&D and manufacturing centers are based in Sweden, Uppsala. And in APAC region, we have our own office in Tokyo and Shanghai. But in Korea, we also have our partner Coma Biotech to support our business in Korea more efficiently. And so actually, as I mentioned before, we are not really a very, let's see, quite new technology in this market. Actually, we have supported lots of customers around the world for more than 20 years. And most of them are regulated labs, like in clinical phase, CROs, and also biopharma industry customers. And many of the top biopharma companies in the world, we have several more instruments installed in their lab or in their lots of different sites. Here is just like we take some representative customers here. The most two customer group we focus on is biopharma and CRO or CDMO companies. So today, because we focus on ADC bioanalysis, but also we could support CDMO with their bioprocessing sample analysis, like for HCP or AAV titer, such a kind of analysis in bioprocessing. We have ready-to-use kit to support. And also we have some other customers in biotech and academia market as well. And also we have a lot of references in global wise and also in APAC wise. We have supported lots of drugs to go on the market. So if you are you have questions on this part, just let me know, because we really support a lot of projects locally as well. And here is our contact information. And in Korea market, we cooperate with Coma Biotech. So feel free to reach out to them for any more information or things. And you could also visit our website as well to learn more about the technology and other case studies or applications.